Good morning, students. Welcome to module 4 of Cell, the fundamental unit of life. In this module, we are going to cover up the following. We're going to study about cell organelles, which are endoplasmic reticulum, which is again of two types, smooth endoplasmic reticulum and rough endoplasmic reticulum. We'll be studying about Golgi apparatus, or also known as Golgi bodies, lysosomes, mitochondria, plastids, and its types, which will be leucoplast and chromoplast, that is the color plastids, vacuoles, which will be plant vacuoles and animal vacuoles. So let's begin. Cell organelles. Now, what is an organelle? An organelle is a tiny cellular structure that performs some specific function within a cell. These are embedded within the cytoplasm of eukaryotic and prokaryotic cells. And in case of more complex eukaryotic cells, you will find them enclosed by their own membrane. That is why they are also called as membrane-bound organelles. As in the first picture above, you can see a prokaryotic cell does not show clearly marked cell organelles, although all of these are present in the cytoplasm and are doing their function. But in case of a more advanced eukaryotic cell, which is showing next in the picture here, you can see that each organelle has its own boundary and you can see it's separate from another or from all other organelles. What you see next in this picture here is called division of labor inside the cell. You can see that each cell organelle is marked with a name and is given a particular function. For example, mitochondria is acting as a powerhouse of the cell. There is a separate cleaning crew. So the one that builds the structure is also assigned a duty. Nucleus is acting as a control center. Endoplasmic reticulum is acting as a assembly line, and so on. So let's study these in detail. Beginning with endoplasmic reticulum. Endoplasmic reticulum in the cell is the transport network of the cell. It is like our roadways. What do we use roads for? We use roads to move from our source, that is from where we live, our home, to our destination. And that is the purpose of a network, transport network in a cell. So what is endoplasmic reticulum all about? It is actually a network of tubes that connect the cell membrane with the nuclear membrane. So like it is connecting the source, that is the nucleus, and the target place. It is made up of two subunits, rough endoplasmic reticulum and smooth endoplasmic reticulum. Where can we find it? We can obviously find it around the nucleus. As you can see in this picture, the blue colored space is the nucleus and around it is a mesh network, which is the endoplasmic reticulum. Endoplasmic reticulum, as we just studied, is of two types, which are rough endoplasmic reticulum and smooth endoplasmic reticulum. What you see in the picture here is a version of this nucleus surrounded by endoplasmic reticulum. It's a cut open picture and you can see it very clearly. The rough endoplasmic reticulum has small dots over it. That is why its surface appears rough. And the smooth endoplasmic reticulum appear obviously smooth. And they are surrounding the nucleus. What is the basic function of endoplasmic reticulum? It works as the transport network of cell, especially for molecules going to specific places inside the cell. But there are different functions for SER and RER. SER is smooth endoplasmic reticulum. RER is a short form for rough endoplasmic reticulum. Let's see what these are. Now, membrane in case of SER is smooth, as you can see in the picture, and they do not contain any ribosomes. Ribosomes are small cell organelles that are also called as protein factories because they are the ones that produce proteins from amino acids. While in case of rough endoplasmic reticulum or RER, the membrane can be seen rough and the ribosomes are present on the surface. That is why it is rough. Also, you can see that SER is made up of a network of fine, smooth tubules. There are no dots over these, no roughness, because no ribosomes are present. While in case of RER, these are made up of little flattened sacs and they have ribosomes sticking on the surface. SER helps in the function of fats or lipid synthesis, while rough endoplasmic reticulum, because of the presence of ribosomes over it, helps in the protein synthesis. In case of some higher vertebrates or animals with spine, The SER helps in detoxification of liver cells, and it helps to detoxify all the poisons and toxins, while RER helps in the formation of enzymes, and enzymes are made up of proteins. So that is all about function of different types of endoplasmic reticulum. I hope you have noted the structural difference between the two. Moving on to Golgi apparatus. This is the packing and the distribution unit of the cell. It is like that warehouse that collects the raw materials, modifies them, packages them into separate packets as our requirement, and then sends them out for delivery. So let's study its structure. It is made up of a series of flattened sacs, pouches, or saucer-like structure, as you can see in the picture. Network of interconnected tubules for the transfer of those packets. Remember, vesicles are the packets, and vacuoles at the peripheral regions. In the cells, Golgi apparatus, in the plant cells, Golgi apparatus is also referred to as dictyosomes. Where can you find them in the cytoplasm? You will find them in the cytoplasm next to ER and near the cell nucleus. In case of plant cells, they are more near to the cell nucleus. As you can see in the diagram, besides the general labeling that I have mentioned in the beginning, cisternae, the flattened sacs, and the vesicles, which are also seen as blue bubbles, some of these are shown cut open. Also in the picture, you can see there is a labeling saying cis phase. Now, cis phase is the receiving phase of the Golgi apparatus, the phase through which it receives the materials, and trans phase is the other phase of the Golgi body. Now, function of the Golgi bodies. Golgi apparatus is involved in the formation of lysosomes, and since we also discussed that it is a packaging unit, so packaging unit is involved in storage and modification of some of these components into the things that we require, into the materials that we required. Golgi apparatus is involved in formation of lysosomes. It is also responsible for the synthesis of cell wall and cell membrane. Remember, the proteins and the lipids that came from the endoplasmic reticulum, they reach this Golgi apparatus, and here the proteins and the fats, that is the lipid, are brought together, modified to form cell membrane, which is made up of lipids and proteins. They also help in formation of lysosomes, as you mentioned, because lysosomes are filled with enzymes, and these enzymes are produced by proteins. So this really is the packaging and modification and distribution unit of the cell. Next up is lysosomes. Since we just mentioned lysosomes, why not discuss them now? Lysosomes are the cleaning crew of the cell. They are also called as suicide bags. They are also called as defense mechanism. They have multiple names because they have multiple functions. Now let's see what these are. These are very simple in the structure. They have a simple membrane bound structure. It is actually a simple small membrane bound vesicle or a bubble kind of a structure, inside which very strong digestive enzymes are filled in. Where are they produced? These are produced by Golgi bodies. We just studied that. And obviously ER has also helped, because ER has helped in the formation of the proteins that are going to change into enzyme and are filled inside the lysosomes. Where will you find these lysosomes? These are found in the cytoplasm. What is the function, which is the most important part? Functions. They serve several functions. Number one, they can serve as intracellular, that is within the cell, intracellular digestive system. Enzymes can also break down the food. So they help in breakdown of food and can help in easy absorption of the food. They can also digest organelles that have outlived their usefulness. So that is why they are also called as cleaning crew of the cell. For example, as you can see in the picture, if some cell organelle has worn out or has become old and is not functioning properly, so a lysosome, as you see at the top of the picture, the lysosome says this mitochondria is not working properly and is worn out and has grown old. So lysosome attaches itself to it, and all the enzymes present in the lysosomes enter this organelle. and then they digest all the contents of this organelle, break down into simpler components, and the small molecules that are produced as a result of it, the broken down small molecules, are released back into the cytoplasm, and any waste from it is released out of the cell. And the useful materials are again used by the new organelles that are formed. So cell is basically using up all of this decomposed matter for formation of new organelles. So this is recycling, cellular recycling. Lysosomes of WBCs also help in destroying pathogens or any foreign particles or bodies that enter a cell, and therefore they act as the natural defense mechanism of the cell or of the cell body. And the most popular function is the one that you already know of. If the membrane of the lysosome happens to get ruptured due to some event, the enzymes of the lysosomes can digest the entire cellular structure, causing the death of the cell, and that is why these are also called as suicide bags of the cell. So let's move on to the next organelle, which is the mitochondria, the energy powerhouse of the cell. Now, each house or each body needs a source of energy. For a cellular body or for a cell, it is the mitochondria. Let's study about it. Mitochondria are cylindrical organelles. These are bound by outer continuous membrane, as you can see in the picture. And outer membrane is continuous, it does not have any breaks, while the inner membrane is porous. The inner membrane is porous and is thrown into folds. These folds are called as cristae. And within this inner membrane, you can see a colored liquid flowing inside it. This liquid or this semi-fluid substance is called the matrix. The cristae actually divide the inner chamber of the mitochondria incompletely, and it is filled with the homogeneous dense material that is the matrix. And the very important thing that makes this cell organelle different from many other or all other organelles is that it contains its own genetic material and protein, which makes the mitochondria not just the powerhouse of the cell but also a semi-autonomous unit, which means it can survive on its own to a certain extent. Where will you find these? You will find these scattered in the cytoplasm. Now, the main function of the mitochondria is to perform cellular respiration. We have studied two types of respiration before. Cellular respiration happens inside the mitochondria. To be very precise, aerobic respiration or parts of aerobic respiration happen inside the mitochondria. This means it takes in nutrients from the cell, breaks it down in the presence of oxygen, and turns it into energy currency of the cell, which is called as ATP, adenosine triphosphate. It also helps to maintain or regulate the cellular metabolism. Cellular metabolic rate needs to be maintained at all the times in order for the cell to function properly. So it is a very, very important cell organelle. Without this, the functioning of the cell becomes nearly impossible. Moving on to our next cell organelle, which are the plastids. They are also very similar to mitochondria. We will discuss about it why. But before that, the structure. Plastids are disc or oval shaped structures. These have double membrane, and they are cell organelles that are found in the cells of plant and algae only, which also belongs to the plant kingdom. So you will not find plastids in animal cells. Plastids are responsible for manufacturing and storing of food. These are often seen containing pigments that are used for photosynthesis, and different types of pigments can change the color of the cell. We can see plants are of different colors. All the colors that appear in a plant are primarily because of these plastids that contain those pigments. Where will you find these plastids? These are seen in the cytoplasm. These are found in the cytoplasm. What you see in the picture is the three basic type of plastids that we are going to talk about today. leucoplast, chromoplast, chloroplast. Basically, all the colorless plastids are called leucoplast, and the colored plastids are called as chromoplast. Chromo means color, plast for plastids. So chromoplasts are colored plastid, leucoplast are colorless plastids. Chloroplast happens to be a type of chromoplast because it contains color. It contains green color. So we are going to study about these in detail next. Moving on to the comparison between leucoplast and chromoplast. First of all, look at the pictures. The first picture is that of a leucoplast of a potato tuber after they have been stained using iodine. And now you know that iodine, when it is put in a starch solution, it turns it into bluish or blackish blue color. So what you see in the cells inside is actually starch granules colored blue-black inside a leucoplast. While the other picture, which appears to be reddish, is of a chromoplast in case of red peppers, red chili peppers. These pictures really look very beautiful. Let's compare the two now, leucoplast and chromoplast. Leucoplast, as we just discussed, are colorless plastids, and chromoplasts are colored plastids, including chloroplasts. So these contain color and they do not have any color. Where will you find them? You will find leucoplast in most of the non-photosynthetic parts of the plant. Non-photosynthetic parts of the plant which are actually not above the ground, which are present below the ground. So underground roots, underground shoots also will be the ones where you will find these leucoplasts. And in case of chromoplasts, all the flowers, the leaves, the aging leaves, the fruits, all contained colored parts, all are actually colored part of the plants. So that is where you will find chromoplasts. Function of the leucoplast is that they act as storage sheds for starch, lipid, protein, all the content that cell produces and needs to store for a long term are put in roots and underground stems. So they are stored in the leucoplasts. And in case of chromoplast, as you know, the flowers are colored to attract insects, pollinating agents. And fruits are colored so that they attract animals or organisms for seed dispersal. So chromoplasts are actually helping in attracting pollinators and agents for seed dispersal. Next up is chloroplast. Chloroplasts are the kitchen of the cell. Chloroplasts are green plastids which possess photosynthetic pigment called chlorophyll. As you can see in the picture above, it shows plant cells containing multiple chloroplasts inside the cytoplasm. Picture below rather shows the structure of a single chloroplast, which is a double membranous structure. You can see the outer membrane and the inner membrane, inside which stacks of coin-like structures are present. This stack is called the granum, and these single coin-like structure along with the pipe-like connecting structure which are connecting this one stack to another stack are called as thylakoids. Thylakoids are the structures that contain chlorophyll that help in the process of photosynthesis. Now, around these is present a liquid or a semi-fluid substance called stroma. So this forms the whole structure of the chloroplast. Now, what does a chloroplast do? Chloroplast absorbs sunlight and uses it with water and carbon dioxide gas to produce food for the plant, which is glucose as a result of the photosynthesis. This glucose can later be stored and in the form of starch after converting it into starch. So therefore, for a plant cell, this is the kitchen of the cell. Do you know that plants are the only organisms, or you can say only beings, that can change sun's radiant energy into chemical energy that can be stored for a longer period of time? We already know that plants are the only things that can produce their own food. But this is what is actually going on. The sun's energy, radiatory energy, is changed into chemical energy by these chloroplasts. Now, the main function of chloroplast, as we discussed, is to produce food during photosynthesis and also to store the food energy. Very important function. Moving on to vacuoles, another storage device for some cells. Now, vacuoles are fluid-filled sacs bound by a single membrane. If you compare the structure with the lysosome, it's very simple to the structure of a lysosome, but it does not contain enzyme inside them. Now, vacuoles of plants and animals are different. Vacuoles of plants are filled with cell sap containing minerals, sugars, amino acids, dissolved waste products. Now, plants need larger vacuoles because vacuoles help them in maintaining turgor pressure within the cells. So, when vacuoles store water, they become turgid with the help of endosmosis or through the process of endosmosis. And when cell loses water, the vacuoles actually help the cell to retain its shape in case of plant cells. While in case of animal cells, vacuoles store nutrients, ions, water. They form excretory vacuoles that help in exocytosis of waste materials. They also form food vacuoles after endocytosis, into which the lysosomes secrete the enzymes. Remember, they help in digestion to help digest the food present in the food vacuole. And bacteria are also sometimes engulfed by the cell and are destroyed in animal vacuoles by lysosomal activity. As you can see in the picture, you can see the visual comparison between the two. We are also going to do a deeper comparison between the vacuoles of the plant and the animal cell, which is here. Now, the vacuoles of the plant are single. Number one, there is a single large vacuole in a plant cell, whereas there are several small, or you could say tiny, vacuoles in an animal cell. First, and the primary difference. Second is that this vacuole in the plant cell is placed centrally. It is present in the center. That is why the nucleus is in the periphery. All of the cell organelles and the nucleus is pushed to the side, and about 90% of the cell space is taken in the center by this one single large vacuole. And in case of animal vacuoles, they are simply, because they are tiny, they are simply scattered in the cytoplasm. What does a plant vacuole do? It stores water and helps to maintain turgidity. And in case of animal vacuoles, they store nutrients, ions, water, as we discussed previously. Plant vacuoles also help to retain the shape and maintain the osmotic concentration within the cell. Remember, we studied osmosis and how it depends upon the concentration inside the cell membrane and outside the cell membrane. So, osmotic concentration is maintained with the help of this vacuole. Animal vacuoles help the cell in endocytosis and exocytosis and also transport of waste materials that need to be excreted out of the cell. So, vacuoles, although small or big, are serving very, very important function within a cell. With this, we come to the basic comparison between, or you can say visual comparison between the plant cell and the animal cell. In this comparison, we are going to see what all things are present in plant cells and that are not present in the animal cells. First and foremost, the central vacuole, which is large, is not present in the animal cell but is present in the plant cells. In comparison, then chloroplasts are present in plant cells, but there are no chloroplasts since they do not need to prepare their food, so they are absent in animal cells. Also, you can see in the diagram that the plant cell contains a cell wall, a very rigid, thick cell wall outside the cell membrane, and it is absent in case of animal cells, which helps us to conclude that the common organelles or features present in both the cells will be mitochondria, ribosomes, endoplasmic reticulum, Golgi apparatus, although in case of plant cells it is called as dictyosomes. With this, we come to end of this module. Happy learning to you.